forums. My name's Peter. I'm part of the forum team that helped put on these events. So we're part of Christian Union. Christian Union's aim is to make Jesus known, and forum is a way that we tackle with the issues in the world and with apologetics and stuff like that. So it's fantastic you could be here no matter what background you're from or belief. It's awesome. So this week we're looking at gene editing, and it's going to be a really exciting talk. Jia-Yee's with us today, and she's got a fantastic background. She's the Director of Industry Engagement at the Graham Clark Institute for Biomedical Engineering. She's done over 20 years of biomedical research, as well as with being ISCAST and being a Christian in science. So let's give her a warm welcome.
Thanks very much, Peter, for the invitation to be here. It's really a privilege to be providing this talk in front of you. Now, as a disclaimer, I should highlight that the views that I be talking through in this particular session are my own personal views and not the views of University of Melbourne. Now, with that out of the way, I just thought I'd gauge in the room how many here are from the biomedical area studying or doing research. Fantastic. Other than Natasha, who else is in engineering? Anyone from engineering? Oh, hello. I'm based at the School of Engineering, so it's Natasha. So good to see a fellow engineer. Let's get started. Well, we are in a very exciting era in medical research. Even though I hung up my lab coat 15 years ago, I still enjoy reading about scientific and medical advances. It's such an exciting area. So we are now in an era of medicine in the midst of genomic revolution. And because we have the 13 billion letters of the human DNA. Now, for the benefit of those who are not familiar with biomedical sciences, this is what we have here is a double-stranded DNA, the DNA helix unraveling out of a chromosome. And the double-stranded DNA is made up of 4 bases or nucleotides. They're called adenine, guanine, cytosine, thymine. They've been abbreviated to these 4 letters, AGCT. And they are the instructions for life, and based on a combination of these letters. Now, the sequencing of the human genome, or the human DNA, was initiated by the Human Genome Project in the US. It was led by Dr. Francis Collins. It was a Herculean effort involving publicly and privately funded research teams, and it started in 1990, concluded in 2003. Amazing effort, 13 years sequencing 13 billion letters of the human genome. When the draft of the human genome was presented to the then President Bill Clinton in the White House, this is what the then President Bill Clinton declared. Today, we are learning the language in which God created life. Now, in this momentous occasion, it's heartening that God is given some credit for this effort. So this is a picture of Dr. Collins. He, in this photo, he has basically declared the successful completion of the Human Genome Project in 2003. When that project started in 1990, I was in the midst of my PhD, so that was a long time ago. Now, if you are interested in learning a bit more about this amazing man, Dr. Collins is actually head of or director of the US National Institute of Health. This NIH is the largest biomedical research agency in the world. Now, I highly recommend this book if you want to learn more about Dr. Collins, because it includes a story of his conversion to Christianity. Now, as a result of this Human Genome Project, we have this sequence of DNA. The scientific and medical community has been embracing a whole bunch of new terms not previously seen before. So we see genomic medicine, personalized medicine, precision medicine, pharmacogenomics. These actually refer to the tailoring of treatment based on the patient's own DNA. It is personalized, it is precise, as scientists and medical teams can now use that information to really customize care, diagnosis and care of that patient itself. Now, there is still a lot we don't know about the machinations of the genome and how it's applied in a biological or scientific context. But we have seen enormous progress and significant advances in the treatment of diseases such as breast cancer, melanoma, and many others. Now, I'm going to dive straight into genome editing because I only have less than 40 minutes now to give this talk. So what is genome editing or gene editing? These terms are interchangeable. So genome editing refers to the use of biological tools that turn on and off genes. So these can be achieved by inserting a gene, removing a gene, or changing one of the 4 letters in the DNA. So while there are several genome editing tools, I'm just going to focus on CRISPR-Cas9. That's because it's received a lot of limelight, and I feel there's a Hollywood script coming up with this narrative of how CRISPR came about. And Science, which is a prestigious journal in the US, in 2015 basically highlighted it was the breakthrough of the year. So it's an exciting tool. It's helped build new cellular animal biological models to better understand disease. It's been applied across the insect world, plant world, and of course in the mammalian and human world. And it's also been used in gene therapy for inherited gene disorders such as haemophilia and sickle cell anemia. So in the application of gene therapy for sickle cell anemia, the US 60 Minutes program actually interviewed Francis Collin, and they tweeted about it. So here is Francis Collin, director of NIH, former head of the Human Genome Project, basically explaining that sickle cell anemia arises because of the single letter in the DNA that is misplaced. A T should have been an A. Now, it's only last week that Francis Collin tweeted his excitement. I'm excited about the potential for CRISPR-Cas9 to cure sickle cell disease. Maybe in the next 5 years, that would involve taking the bone marrow cells from a patient, correcting them with gene editing, making space in the bone marrow, and then putting them back. So where to from here? Well, they're starting to look into clinical trials for this particular disease. Now let's think about what is CRISPR. It sounds like a new brand of potato chips, but thankfully it isn't. CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. Now, I'm glad it's been abbreviated CRISPR. It's a bit of a mouthful. But it was actually discovered by a group of scientists, and it's actually the bacterial's defense mechanism. It's the bacteria's immune system against invading viruses, called bacteriophages. Now, scientists, what they've done is they've hijacked this bacterial immune system or defense mechanism against viruses, repurposed it, re-engineered it, and applied it in the biomedical animal and plant research environment. So just a very simplistic explanation of CRISPR is the Cas9 is a biological scissor. So the double-strand DNA is unraveled, and there's a guide that takes the relevant gene of interest to the right target DNA section. And Cas9, as a biological scissor, cuts double-stranded RNA, and it provides the opportunity for a new gene to be inserted, removed, or a single letter to be replaced. So if you are interested in actually learning more about CRISPR and the precise mechanisms of how it works, there's some fantastic YouTube clips, and I highly recommend you go and have a look at those clips itself. Now, gene editing tools like CRISPR can be applied to edit genes in somatic cells. So somatic cells are just tissue cells. We all have clusters of cells and tissues. And when you apply a gene editing tool like CRISPR to tissues, such as in the case of sickle cell anemia, bone marrows were extracted and they applied CRISPR onto it, those gene-edited cells dies with the patient. By that I mean it doesn't get passed on to future generations. So when the patient dies with that gene-edited cells, that's it. Now, the other challenge of CRISPR is the use in germline cells. Now, germline cells are gene-edited. So they represent the egg, sperm, or embryo, and there's now technology involved using CRISPR on these germline cells. The challenge is editing these sort of DNA in these germline cells means that the effects are then passed on to future generations. And that is a real challenge. So we have the capability to actually modify the DNA of whole genomes with this technology. Now let's go back to the human side of CRISPR. Who are the inventors of CRISPR? And it's a fascinating story. It's playing out as modern-day Shakespearean drama. There's a bit of Romeo and Juliet overtones over this because, now I have to say that when we talk about the inventors of CRISPR, it applies to intellectual property relating to who invented CRISPR and applied it in a mammalian context in human embryos. So let's look at that a little bit closer. There's dispute about the ownership of its intellectual property. So we have the House of Broad in the US East Coast, Broad Institute, led by Feng Zhang, who's the prospective inventor. Then there's the House of UC Berkeley, prospective inventor Emmanuel Charpentier and Jennifer Doudna. Feng Zhang is a neurobiologist. Emmanuel Charpentier and Jennifer Doudna are a microbiologist. Now, what's at stake here? There's a patent war amongst these two US academic giants. And what's at stake? The rights to commercialize products and developments, and products developed by using CRISPR-Cas9 system. A lot of money is at stake here. Now, like Romeo and Juliet, they're not going to live happily ever after. Feng Zhang has been awarded rights to the IP. And the saga will continue, no doubt. So let's then look at the biology of CRISPR itself. Now, in 2017, a team from China tried the technique in embryos, fertilized eggs. So these eggs were taken from discarded embryos or embryos that were no longer in use. And this experiment actually caused a lot of consternation, created a buzz in the international community because they were the first to actually show the viability of embryos that had been gene-edited. So they actually then used fertilized egg taken from sperm of men with genetic mutations that caused anemia. So a couple of embryos were successfully edited. They were correctly edited. Two of the edited embryos had a mosaic effects, which means that the embryos had a mixture of edited and unedited cells. Now, how could that have happened? Let's take a closer look into this. So CRISPR was actually injected into the single-cell stage of the fertilized embryo. The CRISPR-Cas9 edited the gene after the cell most likely had divided and split. So some cells inherited the unrepaired DNA and some had the edited DNA. These types of mosaic effects have been published before. They've been observed in mice and primates. But these embryos are non-viable and they were destroyed because it's worth noting that there's currently a 14-day limit to maintaining embryos in culture after fertilization.
Were they non-viable because they were going to die anyway, or were they non-viable just because of the laws surrounding it?
So in 2015, they published this piece of work in a national journal in China. And so they then published this in 2017 in an international journal, and they were, I think my recollection, it was non-viable embryos.
Because they were mosaics?
They were non-viable because I think what they meant, and I could be wrong here, that in the early part of the experiment they made it deliberately non-viable. Yeah. So a month later, after that publication, a US group also published similar experiments in Nature on gene-edited embryo, this time looking at the correction of gene mutation that causes cardiomyopathy. Now, it's really worth highlighting that both those papers, the researchers in those papers, really flagged the risky nature of the technology. They flagged that more research is required to better understand how to address safety concerns with this type of technology. Now, in the case of the US team, they had indicated that they were able to avoid that mosaic effect of the embryos. Now, the most controversial part of this technology was seen November last year. So at the International Summit on Human Genome Editing, a Chinese scientist called Dr. He Jiankui from University of Shenzhen announced at this particular conference that he had applied CRISPR technology to remove a gene CCR5 in embryos. The embryos were implanted, and twin girls were named Lulu and Nana were born. And now, it caused a huge uproar in the international community. Now, Dr. He wanted to ensure that these babies would be resistant to HIV, the virus that causes AIDS. It looks like he recruited, as part of the study, 8 volunteer couples, males that are HIV positive, females that were HIV negative, and he performed the gene editing on in vitro fertilized eggs. And it looks like that another pregnancy might have been on the way using genome-edited embryos. It's hard to know what exactly happened. These are considered claims because there's no published articles available. So these are the types of international condemnation that have arisen as a result of that announcement. In ABC, for example, headlines are Chinese scientist who claims to have edited twin genes slammed by peers and medical ethicists. BBC reports He Jiankui, China condemns baby gene editing scientist. Now, a prominent Australian bioethicist, Julian Savulescu, he's currently based at University of Oxford, says in his blog, if true, this experiment is monstrous. And there's also a response from scientists from China in an open letter, and this was reported in the ABC based on a Weibo account. I don't have a Weibo account, and I'm not fluent in Chinese, so I'm just going to go by what was being reported by the ABC. It seems like quite a large group of scientists spoke out, and this is what they actually said. Direct human experiments without ethical assistance can only be described as madness. The accuracy of CRISPR gene editing technology and its missed target effect are very controversial in the scientist community. Any attempt to directly transform human embryos and produce babies has massive risks. At the same time, this is a massive blow to the global reputation and development of China's science, especially in the field of biomedical research. So it's a pretty damning kind of assessment of Dr. He's kind of work. Now, I have to note that while there's been a lot of attention on Dr. He himself, there is also the wider question of the team that supported his work. I mean, he wasn't alone. Who ever seen a lab with just a lone scientist in that lab? We always work as a team. And he must have had a medical team to back him as well, because someone had to screen the 8 couples, someone had to implant the embryos and follow the pregnancy through and delivered the babies. So whoever's involved in this, the actual experiment, the wider medical procedure, they were all complicit in this controversy. Let's look then at some of the safety concerns. I'll just see of CRISPR and why the international scientific community is alarmed by what's happened. Germline editing of embryos, as the Chinese scientists have alluded to, have off-target effects. That means that when CRISPR-Cas9 takes in effect in the cell, there are potential unintended DNA changes in the embryo. So, in addition to targeting, being very specific in its targeting, there could be unintended targets as well. We just don't know. There's also the problem, as I've previously highlighted, the mosaic effect. So it turns out that one of these twins has a mix of edited and unedited cells. And if it grows up and they have offspring, this affects future generations as well. So we are truly in an uncharted territories here. From an ethical perspective, we don't know whether there are off-target effects of these twins. We don't know what diseases these twins will be susceptible to now that they don't have the CCR5 gene. And of course, there's the issue of consent when it comes to germline editing that affects future generations. So later generations have no say in what has happened to them. So that's the challenge. So let's go back to the human story and find out, well, let's just see. Right. I'm just going to look at what the context was for Dr. He's experiment before I go back to look at what has happened to Dr. He. So it seems that Dr. He wanted to protect babies from being infected by the father who has HIV. It's a simplistic picture because we really don't have much information other than what's been reported on the internet and in the news. If that's true, the rationale is rather strange. Consider this: HIV is a blood-borne virus that causes AIDS. AIDS is classified as a sexually transmitted disease. Now, safe sex practices combined with now existing very effective treatments of AIDS significantly reduces the likelihood that the mother would be infected and reduces the likelihood that, of transmitting the virus to the fetus during pregnancy. There's also effective treatments after birth for the baby if the mother contracted HIV during pregnancy. So there doesn't really appear to be any justification to edit these healthy embryos. As the bioethicist from the University of Oxford stated, this experiment exposes healthy, normal children to risk of gene editing for no real necessary benefit. And it would seem that Dr. He and his team, or teams, violated the medical ethics of first do no harm. Now, there is another challenge here as well, and the problem is to do with the CCR5 gene that was chosen. It's a gene that produces a protein that helps HIV infect cells. So, rationale would be if you remove this gene, then the babies would be immune to HIV. Now, it's widely known that a small percentage of Northern Europeans do not have this gene. They've somehow evolved not to have this gene, and they appear to be immune to the virus. But one of the challenges, the CCR5 gene that Dr. He used, is not really identical to the one that is missing in the Northern European population. So I would say in this case, near enough shouldn't be good enough because of the repercussions. In terms of the regulations on genome editing, Australia, Canada, Germany, Israel, Switzerland, Netherlands, they banned germline gene therapy in humans for technical and ethical reasons, including insufficient knowledge about possible risks to future generations. And in other areas, in other countries, rather than banning them, they do have guidelines. But the challenge is guidelines are, not really enforceable in some contexts. So it's not clear what breaking these rules might mean. So let's look at what happened to Dr. He. The university, his employer in Shenzhen, basically terminated his appointment. This is what they said, and it was reported in the Xinhua news agency. The scientist executed his experiment on his own. So the university has distanced itself from the doctor. And he did it in pursuit of personal fame. So the motives were questioned. So what has happened to the scientist? It appears that he's in a heavily guarded university-owned apartment in Shenzhen. This was reported in the New York Times late last year, and apparently friends have verified that person in that balcony is indeed the scientist, Dr. He. So that's Dr. He himself. But what has happened to the twins? Well, we just don't know. There is no news about their whereabouts. There are lots of questions being asked, but we don't have any answers. So let's just look at going back to the International Summit on Human Genome Editing. These are made up of esteemed researchers in the field. And these are some of the things that the statements have made. Many questions remain about science, application, ethics and governance of human genome editing. Of particular concern is the possibility of heritable genome editing, which would alter the germline and applications for purposes other than to treat disease or disability. So I think that the voice of the international community is very strong. And in this instance, the scientific progress and the advancement has really outpaced societal consultation and outpaced our discussion on the ethics of germline editing using embryos. So we've heard from the international community. Now let's look at what God has to say. Now, the first few chapters of the book of Genesis tells us why we live in an imperfect world where there are diseases, pain, and suffering. So in Genesis 3, we see that despite God's provision for humankind, they wanted to live on their own terms, not on God's terms. So God had prohibited humankind to eat the fruit. There were two trees that was a no-go zone. So one was the tree of knowledge of good and evil, and he also prohibited humankind from taking the fruit from the tree of life. But humankind chose to ignore this prohibition. They went ahead and ate the fruit from the tree of knowledge of good and evil. The underlying motive here was that they wanted to have wisdom, to act like God. Now, this was, I'm sure, would have been upsetting for God, and he would have looked at, what next could these human beings be doing? He could foresee that there was a likelihood that humankind would also eat the fruit of the tree of life, and the consequences would be that humankind would be immortal, like God. So what did God do? He expelled them from the Garden of Eden. Humankind paid a very high price for ignoring the prohibition imposed by God. They paid a high price for their disobedience. Now, as a result of being expelled from the Garden of Eden, there is now a chasm between humanity and God. And outside of Eden, humanity would experience pain, suffering, and be exposed to a range of diseases, genetic mutations. I think the good news is that we see throughout biblical history that God never abandoned humankind. He always had a plan to reconcile humankind to him. And so Jesus Christ is almost God's version of CRISPR, being to fix the problem. So unlike CRISPR, Jesus is perfect. No off-targets here, no mosaics happening. In being fully God, fully human, he showed us his divinity, and as well as his humanity. He acted with compassion, he acted with mercy in healing many with their diseases. Jesus was purposeful and he was targeted in his mission. So in the gospel accounts there are many instances of Jesus healing, and when he heals, Jesus was actually pointing to the new heaven and a new earth where death will be no more, mourning and crying and pain will be no more. And this description can be read in Revelation chapter 21. So God gives us real hope in Christ as we live in this imperfect world. Now, what does God have to say about conducting medical research and also using that on embryos? So I'm going to just first explore what it means to be created in the image of God. So Imago Dei means the image of God. And this is what Genesis chapter 1, verse 26 to 27 says. Then God said, "Let us make man in our image, after our likeness, and let them have dominion over the fish in the sea, and over the birds of the heavens, and over the livestock, and over the earth, and over every creeping thing that creeps on earth." So God created man in his own image. In the image of God, he created him. Male and female, he created them. So it's a big statement. But what we can glean from that is that it shows us that we have a special relationship with God. We share his moral, spiritual, and intellectual nature. And we share God's desire to care for humanity and for the world, to act as good stewards of God's created world. Now, the Apostle Paul, he wrote a letter to the Ephesians, and in that letter he states, "For we are God's handiwork, created in Christ Jesus to do good works, which God prepared in advance for us to do." So I would affirm that medical research can be seen as good works that we should do. But the question is, are there any restrictions for Christians using tools or biological tools in medical research? And I would say yes, that we need to be cautious. Now, the reason behind that is to do with the embryo. There's much debate on the use of the embryo in research. It's important to engage in this debate, and it informs our decision on whether embryos should be experimented on. The issue at hand is the moral status of the embryo. Is the embryo human? Well, it's certainly human. It's not a dog embryo. It's not a cat embryo. But the bigger question is, does the embryo bear the image of God? Does the embryo have worth and value? And this is the challenge that we have to wrestle with. And different Christians hold different positions on this. Now, in the scientific community, I do need to point out, there is a 14-day rule for experimentation of embryos. Scientists can only experiment on embryos up to 14 days of fertilization, at which point the embryos must be destroyed. The 14-day was chosen because day 15 is the emergence of what we call the primitive streak, a precursor to the brain and the spinal cord. So the 14-day rule is law in 12 countries. That makes it a criminal offence to keep human embryo alive in a laboratory or in culture for longer than 14 days from the time of fertilization. So Australia and New Zealand have this law in place. Now, some in the scientific community would argue that the embryo is a collection of cells, and they would therefore say, okay, let's experimentation on embryos up to 14 days is permissible. But I do want to stress that such demarcation on embryonic development is artificial. Embryonic development is a continuous, ongoing process. And if we take a viable embryo in culture and implanted it, the embryo would develop into a human. After all, we all start at life as embryos. My memory's not too good on that one. But I also want to highlight that the psalmist gives us some perspective as well. He stands in awe, recognizing that God formed his inward parts. God knitted him together in his mother's womb. The psalmist acknowledges that he is fearfully and wonderfully made. So in this context, the embryo is not just a collection of cells, and I believe that we should be cautious so that we do not treat embryos as a commodity to be experimented on. Now, I do want to then move on to talk about the use of CRISPR in the enhancement of human traits. Now, there's a program on the US PBS show called It's Okay to be Smart. It's on YouTube, and it cleverly poses this question. Does CRISPR make people better, or does the technology make better people? Now, who's seen Gattaca? It's a 1997 movie. Oh, great. I love Uma Thurman, Ethan Hawke and Jude Law. They're some of my favourite actors. I'm a huge sci-fi fan as well. So I loved this movie when it came out. It's also showing my age. Now, Gattaca, the film is named using 4 letters of the DNA. And it portrays a time, apparently 1997, bear in mind, it's where it says in the not-too-distant future, children will be ordered to design based on the traits preferred by their parents. That is, if you can afford the technology. So in the show, the challenge posed and experienced is that there was societal discrimination based on one's DNA pattern. So those born, there's a eugenics program, those born within that eugenics program were referred to as valids, and then those born through natural means, that means they were susceptible to genetic mutations, susceptible to this whole range of gene disorders, they were called invalids. So critics of the CRISPR technology, they're reminded of this movie and are concerned that the technology could be used to enhance traits that would affect future generations. We would be genetically engineering a population of sort of human beings with advanced traits. So genetic enhancement could mean smarter babies, one that performs better in sports, and so on. Now, this is rather simplistic because genetic engineering traits is much more complex than doing gene editing on single genes itself. But it does beg the question, who has the right to declare what is good? Who has the right to declare what is a good trait and what is an inferior trait? Well, I would say that this is in God's domain. I would say that the Bible tells us in Genesis 1, we read that each time God created, he declared it was good. And in Genesis 1, chapter 1, verse 31, this is what was said. God saw everything that he had made. God saw everything that he had made, and indeed it was very good. Everything includes the creation of humankind. Now, I'm noticing that time's kind of slipping away, so I'm just going to sort of move to what's wrapping up and focusing on what it means for me as a Christian to live a Christ-centered life, how it's impacted my career as I've made some difficult choices. The use of embryos for research was a personal challenge for me when I was a young postdoc. I had developed, while I was doing, this was in my first postdoc, I had developed a primary cell culture technique when I was doing research on hepatitis B virus. So my background, I'm a research virologist. And my supervisor was very, very keen for me to actually go and work in a lab in San Francisco, where my skills in this area could be applied. And the challenge was that this US team was working on embryonic stem cells. And working on stem cells derived from embryos would mean that the embryos would be destroyed in this process. Now, as I hold the belief that embryos have the capacity to become human, I was really not comfortable working in the field. So there was some tension on whether I should go or shouldn't go, and I ended up deciding not to take up what was supposed to be an amazing opportunity, much to the disappointment of my supervisor, as he believed that it would have been a great career move for me. It would have been great for him as well, because he suggested the US lab as a potential opportunity for bridging closer ties with collaborators in the US. Now, another challenge that I faced was when I felt the weight of responsibility leading an NHMRC-funded research. My PhD was on rubella virus, and I was very blessed to have NHMRC fund my work on rubella virus after I graduated from my PhD. So I recognized that it is very simple and very easy to join various pieces of viruses that we were working on. And I had to repeatedly remind my honours and PhD students of the dangers of stitching together these viral genomes, because it is possible that in error we may have created viable superviruses that could potentially have disastrous consequences. So my attitude was, just because we can use the technology doesn't mean we ought to. So we have to be very careful, we have to be cautious. So it's important to be aware of the implications of research and really to apply a Christian framework by which we make decisions. Now, while I no longer work in research, there are societal implications in using gene editing that I need to be cognizant about. I do have a responsibility as a layperson to actually speak out if I feel that there are violations of Christian principles. In some instances, I speak out at the ballot box. Especially, I certainly will be speaking out in the up-and-coming election. And that's happening in May 18, by the way, if you haven't caught up with the news. So, to summarise, I think we can be really thankful for the progress that exists. We can be really thankful for the Human Genome Project. I would encourage us all to engage in debate on the use of embryo in research and the role of germline gene editing. It's important to understand the Christian framework and the principles that supports our position. When the scientific community puts out statements or guidelines on aspects of medical research, it's really important to understand why you agree or don't agree with these positions. And I'd encourage you to examine your motives when you do your medical research and when you conduct your experiments. And I'd encourage you to be aware that God continues to have a voice in this whole area. For Christ is seated at the right hand of God now, ruling over this world. God will continue to have a voice. So I'd just like to finish up by leaving you with John chapter 1, verse 1 to 4. In the beginning was the Word, not the genome. And the Word was with God, and the Word was God. He was God in the beginning. He was with God in the beginning. Through him all things were made. Without him nothing was made that has been made. In him was life, and that life was light of all humankind. The light shines in the darkness, and thankfully darkness has not overcome it. So at this point, I'm happy to take questions. Thank you.